Hi, welcome to my video on DNA structure and replication. What is DNA? For those of you who may not know anything about DNA, DNA is short for deoxyribonucleic acid. It carries genetic information in all living organisms. It has a double helix structure with 2 strands. Most of the time, DNA will be found in the cell nucleus. Now that you have a little information, I will begin talking about the structure of DNA. DNA is made up of molecules called nucleotides. Nucleotides are made up of sugar, a phosphate group, and a nitrogenous base, such as adenine, which is shown in the picture, and also guanine, cytosine, and thymine. Nucleotide bases come in 2 groups, which are purines and pyrimidines. The purines are adenine and guanine. The pyrimidines are cytosine and thymine. Complementary base pairs. Nucleotide bases are linked by a hydrogen bond on opposite strands. They come in pairs, such as adenine and thymine and cytosine and guanine. As shown in the picture, adenine and thymine are together, and guanine and cytosine are together. Lastly, of our DNA structure, we have our primes, 5' and 3'. These are the guidance for the strands. They show the direction. 5' is the phosphate groups and 3' is the carbon A hydroxyl. Now that the structure is out of the way, we will begin talking about DNA replication. DNA replication occurs in mitosis and meiosis. In mitosis, it happens during the cell reproduction, and in meiosis, it happens in gamete reproduction. The start process of replication begins with unzipping the double helix, which is shown at the beginning of the picture. Then the enzyme helicase breaks the hydrogen bond, and this causes the complementary pairs to separate. And now this leaves us with a leading strand, which is continuous, and also a lagging strand, which is discontinuous. Next, we have what happens in the leading strand. What occurs next in the leading strand is the RNA primer binds to the leading strand, and DNA polymerase also binds to the strand. Soon after, new bases are being added to the original strand, as shown in the picture. See how the new strand is being added, and now we have our new complementary bases. Next, in our lagging strand. In the lagging strand, RNA primers are being made, and they start to bind to different parts of the strand. Then the Okazaki fragments, also known as big chunks of DNA, are added to the strand. And in the picture, you can see that the Okazaki fragment is being added to the lagging strand. Lastly, in this part, the complementary bases are starting to be paired, and then they're being checked for any errors that may have occurred during this process. After that, the DNA ligase seals the sequence. This is the ending of the replication process, but not the actual results. The results is each molecule will have a new and an old strand, which makes two daughter DNA molecules, and the DNA double helix will start to form automatically. This is the semi-conservative outcome out of the three. With the semi-conservative, there is also a conservative and a dispersive, which is shown in the picture. And also, as you can see, the conservative and the dispersive has a different outcome from the semi-conservative.
DNA replication is
Lastly,
defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Thank you for watching this video. It may be used as an example to anyone who would like to do so. Thank you.